20 nanometers, very small in size, very 20 nanometers to around the 450 nanometers are there.
Page
There is
number?
no, so here page number 29. Okay, first of all you see this, you listen this some topics and see the characters. Okay, so very small in size and remaining cell organelles are absent. Remaining cell organelles are absent. Only the DNA, either RNA, only the nucleoprotein is present. And that nucleoprotein is covered by the envelope is there. That is the, we call as a capsid or coat, which is the made up of the protein. Okay. So then, so this is the characteristic features are there. So next one is the, here this protein, this virus is made up of the which is differentiated into two parts are there, which is the nucleic acid, another one is the coat, that is the protein capsid. So based on the here, based on their characters, based on the presence and absence of the these protein capsid, here virus are two types are there, that is the nucleocapsid virus. So nucleocapsid virus means here nucleic acid is present, then which is covered by the capsid is also. So only the virion virus. Virion virus means only the nucleic acid is present. There is no capsid. There is no envelope. Okay, nucleic acid capsid. That is the nucleic acid. Nucleic acid means both are present. Capsid and nucleic acid is there. But this virion here only the nucleic acid is present. There is no sheath. So another one is the structure of the virus. Here the structure of the virus here, this having the helical structure. Helical structure is there, and the some. So this is the helical structure is there. So helical structure means this virus is helical symmetry is there. So helical symmetry is there, which is having the a protein port is there. That protein port is made up of number of subunits are there. That subunits we call as a capsomeres. So number of capsomeres is together to form a capsid layer is there, which is the which is surrounded the nucleic acid. So they can see on the electron microscope only. So these icosahedral symmetry, these icosymmetral polyhedron is having 20 equatorial triangular faces and 12 vertical layers are there. Okay, so helical structure is there, and another one is the isocahedral symmetry is there. Two symmetries are present in the viruses. Okay, so another one is the so what is the function of this capsid? What is the main function of capsid here? To protect the nucleic acid. Protect the nucleic acid and to introduce these nucleic acid into the host cells. Then enter into the host cell, and sometimes which is the stimulated from the antibiotics. So what are that they can produce with the antibodies? You know that. And when the host is take the antibiotics, inside the antibiotics, some antibodies are produced. So these capsids they can stimulate the against the antibodies. Okay. So another one. So some the next one is the reproduction of the virus. Okay. So how they are reproduced? So we already discussed that the only the virus can reproduce in the host cells only. When they are entered into the cell, they can control the virus. The host, all the cell organelles are controlled by the virus. When the virus is entered into the host cell, all the activities is controlled by the virus only. So the virus is mainly dominate their character. Okay. So here mainly the life cycle of the
Page
virus.
number?
So the life cycle of the virus here mainly 34. So here 6 stages are there. In the 6 stages are there in the life cycle of the virus. First one is the attachment, penetration, uncoating, replication, assembly, and finally release from the host cell. These 6 stages mainly we can observe in the virus reproduction. So, what is the first one is the attachment. So, in attachment, the virus is okay. So, the virus is uh, attached to the host cell, surface of the host cell, attached to the surface of the host cell. Means, so first of all, to identify the host cell, and then observe the specialized receptors are there, receptor sites are receptor places are there. Then that places the virus is settled down. So after that one, after settle, they are to penetrate the virus into the host cell. Penetration. So the flexible cell membrane of the host is penetrated by the whole virus or its nucleic acid. What happened here? The virus is penetrate into the host cell, maybe enter into the whole virus or only in the nucleic acid. So here mainly some enzymes are synthesized by the virus. So this lysis is observed here, means endocytosis. So the entire virus is engulfed by the cell and enclosed a vacuole or vessel. The viral envelope can also directly fuse with the host cell membrane. So during the penetration period or stage, what happened here? Some enzymes are released by this virus. They can they penetrate the cell membrane of the host cell. So after that is the virus is the capsid or envelope is associated with the host cell wall. So next one is the after the penetration, then uncoated. So here uncoated. What happened here? The enzymes, there are some enzymes and vacuoles dissolve the envelope and capture the virus is now uncoated. So host cell means host some enzymes are there. Then vacuole some dissolved solvents are there. This with help of this one, the virus is enveloped is dissolved. Only the nucleic acid is present. So that one is the uncoated virus. So next one is the synthesis. So after the so here the show the diagram is there. I shown the diagram here. So here see here. So first of all, the virus is entering the host cell membrane, and then gradually it is digest the envelope of the virus. Only the nucleic acid is present in the host cell. So these nucleic acid is then synthesized their uh, RNA or DNA. Okay, RNA or DNA. Mhm. So what is the synthesizer? Synthesizer is nothing but replication or synthesis amoeba. So the virus is involved the primarily multiplication of the genome. Means their genome is copied. Duplicate genome is synthesized by the virus. The replication involves the synthesis of virus messenger mRNA from early genes, and the viral protein synthesis possible assemblage of the viral protein. Then the viral genome is replication mediated by the early or irregular protein expansion is there. So what happened here? Some early genes, some early genes are there, so that early genes can help in the synthesis of these nucleic acid. That is maybe RNA or DNA, anyone. So after the synthesis, so here number of assemblies are assembly means forming group. So here assembly. So after synthesis of the nucleic acid, they can a mature virus. So a well-developed virus are to gather or to form a group. So in this one here having the cell means envelope is there, then nucleic acid is there. So after that stage, after complete or mature the these virus, so release it to the from the host cell. So these release means the cell membrane is lysis, with help of the same enzymes are produced by the virus. Means the lysogenic enzymes are there. Means lysis, cell membrane is gradually lysis. It will breaks down or bursting with help of that enzyme, which is synthesized by the virus only. So after, so what happened here? The cell membrane is gradually burst or killed. Some viruses undergoes to lysogenic life cycle, where the viral genome is incorporated by the genetic recombination into specific place of the host chromosome. So here mainly the virus is replication by here two methods are there. First one is the lytic life cycle, another one is the lysogenic life cycle. Lytic life cycle and another one is the lysogenic life cycle here. So, for example, we can see the bacteria life cycle, lytic life cycle, and another way is the lysogenic life cycle. Two ways are there. So, in lytic life cycle, what happened here? The bacterial genome means the virus genome. The virus genome is not associated with the host cell, host genome. The virus genome is not associated with the host genome, means that is bacteria or plant or any other. Okay, it's not associated. They are individually synthesized and to release it to the virus. So another one is the lysogenic life cycle. This lysogenic life cycle is what happened here. The virus genome is associated with the host genome. The virus genome is associated with the host genome. So what happened here in this lysogenic here the some recombination is occurred. The recombination is occurred. The recombination is occurred.
Please, anyone say page number?
The page number here. That is the page number 36. Okay. So only in the lysogenic life cycle here we can see the recombination. So recombination in what happened here? Some chromosomes are exchanged. The DNA exchanged in the host cells, means that is the bacteriophage or fungi, any other. Okay. So the viral genome is known as a provirus. In the case of bacteriophages, as the four phases, that we call as a provirus or in the prophase. So here the two methods are there: lytic life cycle. Another one is the lysogenic life cycle. Okay, only the recombination is occurred in the lysogenic life cycle only. Okay, so next one is the transmission. Transmission means spread, transmission of the virus. So spread from one host to other host, or one whole organism to other organism by the some different types of the vectors are there. So that vectors is aphids are white flies, hoppers and beetles, nematodes and mites and plasma diploids are there. Different agents are there. Vectors are there. So first one is the through sap. The virus can be spread by the direct transfer of the sap or contact the wounded plant with a healthy plant. So through the sap means what happened here? The wounded parts, wounded plant parts may be contact with the healthy plant. So that way the directly to spread in the these sap. So another one is the insect. So plant virus means the plant virus can transmitted by the vector most of these loop hoppers and some white flies also. Different types of the bacteria viruses are. For example, tomato spotted wilt virus, which is the transmitted by the some trips are. Okay, next one is the nematode. So through the viruses can also transmitted through the nematode. So soil-borne nematodes also prone to the transmit virus. The virus can be transmitted both
Sir,
non-persistently or persistently, but there is no evidence for the virus being able to replicate in the nematode. Okay,
sir,
so another one,
please speak louder.
loudly. Okay, okay.
Yes, sir.
So another way is the then seed and pollen-borne viruses are through the seed and pollen-borne viruses. So from the diseased seeds, so the plant is diseased by the virus. So in that time, what happened here? The in the time here, the virus is from plants to seeds. They can carry from one generation to another generation. So another is the pollen tube is also pollen-borne diseases. So if the pollen grains is also carry these virus. Okay. So another one is the that is from the human human beings. Human beings also a agent as a transmitter of the virus in human beings also. So this is the reproduction virus classification and virus means morphological features. Another one is the reproduction system and transmission. So another one is the some important viral diseases. What are the viral diseases are there? So here, these tomato, for example, some virus diseases are there. Citrus virus are there. So this virus is via aphids. The spread through the aphids are there. So the tomato spotted wilt virus. The tomato spotted wilt virus. What happened here? The symptoms are appeared on the plant leaves, which is the brown or yellow spots are there, and the leaves will be wilt. So another one is the cucumber mosaic virus. So
Yes,
cucumber
sir.
mosaic virus here and here the leaves will be some decoloration are there, and it will be also after some spottings are there. Okay, next one is the rose mosaic virus. Rose mosaic virus also leaves will be decoloration, which is the white and yellow spots are there. So different types of the viruses are there. So they cause the different diseases are there. Potato virus, cauliflower mosaic virus. Okay, so the barley yellow virus are there. Okay, different types of the viruses are. Okay, so here from from the virus here mainly some important questions. What are the important questions here in the virus structure of the virus and reproduction of the virus, and another one is the transmission of the virus. These three topics are in exam point of view, which is important. Okay, Nandi. So next one is
Sir,
the
once again, you can repeat that question, sir.
structure of the virus.
Yes, sir. Next.
So structure of the virus, then reproduction of the virus, replication of the virus.
Next, sir.
So another one is the transmission of the virus.
Only three, sir.
Sir, reproduction of the virus. Next.
Transmission of the virus. So what are the vectors
Plants?
are there? Transmission of the virus. Transmission of the virus. Okay.
So
Here
this will be coming in the essay question, sir.
Some not sure. Maybe some expected questions. Okay. Some expected questions are there. Okay. So next one is the clear. Okay. So next
Yes,
one
sir.
is the cyanobacteria. So here cyanobacteria means the cyanobacteria in family we call as a blue green algae BGA. Okay. So what is the blue green algae? So family we call as a blue green algae. So these blue green algae we call as a the prokaryotes. BJ is also cyanobacteria is also considered or coming from the blue prokaryote. Here not
Page
developed
number?
cell organelles. The not developed cell organelles. This one. Okay, 69. Not developed the cell organelles are there. So here there are unicellular. The cyanobacteria here unicellular are there, multicellular, uniseriate, or some colonials also formed. Very small in structures are there, very unicellular are there. Okay, after they can grow as a colonist means multicellular types are there. So here chloroplast is also present. Then they can prepare the own food. They can live in the fresh water only. The cyanobacteria they can grow in the fresh water only. Means lakes, streams, ponds, fresh water only. So as well as the terrestrial, wet terrestrial means wet areas where the wet areas is present. The cyanobacteria they can grow on the wet areas.
Hello, sir. What happened?
Hello. Hello. So.
Hello, sir.
What happened? Not clear. Hello. Hello.
Sir, you skipped unit 5, sir. Bacteria. Sir,
Not
we
clear.
are directly studying cyanobacteria, sir. So bacteria is not important, sir.
What?
Unit 5, sir.
Bacteria is completed.
Okay.
Okay. Bacteria is that last class is completed. So what happened here? They are the remaining cell organelles are not developed. Remaining chloroplast chlorophyll A and these present. Some mitochondria is also there, but not developed as a eukaryotic character. So these cyanobacteria can play in the vital role in the nitrogen fixation. Many main role in the nitrogen fixation. So these atmospheric
Hello, sir.
nitrogen. What happened? Hello? Hello? This is clear? Okay.
Yes,
So,
sir.
so here atmospheric nitrogen. The free nitrogen then can fix the as ammonia and nitrate with help of these BGA, blue green algae. For example, NASTA. Okay, so NASTA is mainly nitrogen fixing agent. So some blue green algae are associated with the other plants are there. So they can show a symbiotic association in the some angiosperms. For example, there is the Anthoceras. Anthoceras is having the NASTA. Okay, so with help of these nitrogen fixing agent, they can replace it by the ammonia by the plant. Okay, so the cyanobacteria is known as the prokaryote. We already discussed that one. Here, prokaryote characters are there. So next one is the what here. This cyanobacteria is covered a mucilage sheath is there. Cyanobacteria is covered, the total cell wall is covered a mucilage sheath, gently mucilage sheath is there. A mucilage sheath is present. So all cells are some single cell or either some multicellular filamentous like structures are there. They can form the long and straight chain like cells are there, some branching cells are there, some filamentous forms are there. So within some cells are modified as a heterocyst. Some cells are modified as a heterocyst. Okay, modified as a heterocyst are there. With help of these heterocysts, here the nitrogen is fixed into the salt. Okay, so the cell structure of the cyanobacteria. So we discussed this is a prokaryotic cell. We know that one. This one is the prokaryotic cell. So that's why all characters are similar in the prokaryote. So means here the incipient DNA is present. Incipient DNA means not developed DNA. Not developed DNA means here the nuclear membrane is not present. Nuclear membrane is not
present.
Huh? What?
Nothing, sir.
So nuclear membrane is not present. The remaining some cytoplasm is there. So some cells are modified as a harmogonium. So this harmogonium is participate in the reproduction. Okay, the harmogonia they can involve in the reproduction. And remaining mitochondria is present. 70 types of the ribosomes are present, and some cytoplasm is also there. Vacuole is also present. Okay, so this is the structure of the cyanobacteria. What are here? The some chlorophyll A and phycobilins and phycoerythrins is also there. So these means cyanobacteria is mainly involved in the nitrogen fixation. As the symbiotic relation is there, some anthoceras like that. They can exhibit a free nitrogen fixing agent is also present. A specialized cells are there, that is the heterocyst. Heterocyst, a specialized cells are there which is involved in the nitrogen fixing. For example, Anabina, Nastak, Oscillatoria. These are the nitrogen fixing agents. So another one is the reproduction of the cyanobacteria. The cyanobacteria is produced by the vegetative and asexual reproduction. Here, sexual reproduction is totally absent. Okay, so the vegetative reproduction here reproduced by the vegetative in the fission and fragmentation, and hormogonia is also formed. So in the fission, what happened here? In the fission, the fragment, hard chain-like structure that will be breaks down into small fragments, small pieces. That each fragment again they can develop as a new thallus. Next one is the hormogonia. So this hormogonia is the specialized structure of the hormogonia is present, which is involved in the reproduction. So in the cytonema, in the oscillatory or cytonema, so this is the hormogonia is there, very thick cell. Like this is the hormogonia. Again, I show the another diagram here. This one is the hormogonia. So means that hormogonia means the cytoplasm coming from the fuse, okay, coming from a side. So that one is a small fragment will be breaks down from the mother thallus. That hormogonia is again they can exhibit as the independent. They can produce the new cell division and produce a new thallus. So in asexual method. So the asexual method is produced by the actinides. Actinides and endospores. Actinides and endospores. Another one is the exospores also. So this is the actinide formation. Actinides is they can produced in the unfavorable conditions only. In the unfavorable conditions only, the actinides are formed. So unfavorable conditions means in dot areas or some alkaline nature or acidity nature, alkaline nature or acidity nature. When it is to resistance the some unfavorable conditions, resistance from the unfavorable conditions, the actinides are formed. Okay, so in the actinides, what happened here? Any intercalary cells or terminal cells. Any cell of the intercalary cell or terminal cell of the thallus, that is the having the very thick cell wall, and dense cytoplasm. They are going to the resistance period or
Sir,
going
once
to
again repeat it, sir.
some
Once
Hello?
again repeat it, sir.
so
Hello,
any
sir.
cell of
Once
the
again repeat it.
any cell of the intercalary cell,
Asexual reproduction, sir.
asexual
Asexual reproduction
reproduction.
is finite.
In the asexual reproduction, here some actinides are formed or endospores. So the actinides are any cell of the intercalary cell or terminal cell. They can formed as a actinide, which is covered a very thick cell wall, and dense cytoplasm. So these actinides are resistant to the unfavorable conditions. Means dot areas are acidic nature and alkaline nature is there. They are resistant to the unfavorable conditions only. So what happened here? The very thick cell wall is there. Due to the very thick cell wall, they can possess their resistance character is there. High heat or UV rays from UV rays, from high heat, high temperature, and acidic nature and alkaline nature and dot areas, dot character. From these conditions, the unfavorable conditions only the actinides are developed. So another one is the endospore. So in the endospores also formed. These endospores are in the favorable conditions only. This is also actinide picture. So this is the actinide diagram. In the endospores is also formed in the favorable conditions only. So what happened here? The in favorable conditions, the cytoplasm of these cyanobacteria into mitotic division. It is breaks down number of mitotic division, and that small cytoplasm they can exhibits as a new plant body. Okay, a new thallus. A new thallus. That is the endospore. So after a complete development of the endospore, the thallus wall will be breaks down and then released to the environment. Okay. So this is the endospore. So another one is the some exospores is also formed. These endospores and exospores are formed in the favorable conditions only. Okay. So the life cycle of these cyanobacteria here is vegetative reproduction and sexual reproduction. Vegetative reproduction and sexual reproduction are there. Okay. So in sexual reproduction here, the formation of the actinides and endospores and exospores are. So we can see these some type studies are there. That is the nostoc. So this nostoc is the Nostocaceae family. The order is the Nostocales. The family is the Nostocaceae. So these nostoc they are looks like a spiral chain, beads chain like structures are there. Each cell is the egg shape, spherical in shape. Okay. So number of cells are formed a chain like structure. So each cell is covered a mucilage sheath, gelatinous sheath is there. So nostoc they can found in the only fresh water only, mainly fresh water. That is the means streams and ponds like that. Okay, so here they can the filaments means very single cell. Nostoc is the single cellular is there. That is they can form a spiral like structure, beads chain like structures are present. Multiple chain like just like a chain like structures are there. Okay, so here they are to cover a very thick gelatinous or mucilage sheath is there. So inside the cells here some phycobilins are there from chlorophyll A and chlorophyll B is present. Some carbohydrates are there. Okay, so the show here the page number 76. So here you see this nostoc picture, the diagram here. See that a chain-like structures are there. So inside some heterocysts are present. Some intercalary cells they can form the heterocyst. So these heterocysts they can participate in the nitrogen fixation. Okay, they can reproduce by the fragmentation. Nostoc they can reproduce by the small fragments are there. fission or fragmentation. Similarly, actinides also form, but sexual reproduction is totally absent. So another one is the cytonema. This cytonema is also belongs to the Nasticales order and Cytonemaceae family. This cytonema is a cylindrical like structures are there. A filament, cylindrical filament like structures are there. Okay, they can also grow on the fresh water. They can grow on the fresh water or aquatic water, either a free floating agent. So sometimes they are to grow on the terrestrial rocks, wood and soil or plants also. So this cytonym is also the fix the nitrogen. This is also they can fix the nitrogen. So these filaments are very dark and tough among the other algae. So these cytonema, we can see the false branching is present. Cytonema, we observe the false branching is there. So what happened here? A mucilage sheet is there. So due to the mucilage sheet, the filament will be break apart at the site of the dead cell. So what happened here? From the when the filament is break apart at the site of the dead cells, which looks like a branching. Pseudo branching is appeared in the cytonema. Here also cytonema is also having the chlorophyll A and phytocyanin and phytoerythrin is also present. Some pigments is also there. Okay, so they can reproduce by the asexually and vegetative reproduction. Asexual and vegetative reproduction by the in the reproduced of the cytonema. Okay, Nandi. From this one, okay, from this one here only some important questions are there. Okay, some important questions from the cytonema here, cyanobacteria here, the general characters of the cyanobacteria. Okay, Nandi. Hello.
Yes, sir.
Some cyanobacteria here, general characters of the cyanobacteria.
Hello, sir.
General characters.
Sir, please
General
say
characters
again.
of general
Sir, please
characters
say
of
again.
the general characters of the cyanobacteria. It will coming on the examination. Some important question. This one from the cyanobacteria.
Sir, can I get your WhatsApp number?
Okay, Nandi. Huh?
Your WhatsApp number.
చెప్పాను కదా మొన్న చేశాను కదండి గ్రూప్ లో చూసుకోండి మొన్న చేశాను ఓకే ఆల్రెడీ సెండ్ దట్ వన్
Sir, only 2 questions.
Excuse me, sir.
From the virus side here.
Yeah, general bacteria or the cyanobacteria or
okay, from the virus and cyanobacteria here we are completed today. Okay,
Yes,
from the virus here, structure of the virus and replication of the virus and transmission of the virus. These 3 questions are important from the virus. From the cyanobacteria here, general characters of the cyanobacteria.
Excuse me, sir.
What?
Sir, I am not there in WhatsApp group, sir. Please add me. I am new, sir.
Okay, you can create that one. I am not created that one. You can your friends are you can create yourself only.
No, sir. Actually, I am from distant learning, sir.
Okay. Okay. So.
Sir, please say again questions important.
Again, how many times?
yeah.
Sir, please explain.
One more last time.
Okay.
One more. Say fast, sir.
Structure of virus, reproduction of virus, transmission of virus, general character of cyanobacteria.
ఇప్పుడు తెచ్చారు.
Hello.
Sir, are you there? He is not there. I think he is
कोई
left.
बनाए तो मुझे ऐड करो ना कोई भी.
Anyone share group link in chat so that they can come? Alha, are you there? Can you share group link here so that newcomers can come?
Please share करो, मैं WhatsApp join कर.
Hello.
Can anyone send important question in the group?
Hello.
Yes.
Hello, sisters and brothers. Hello, sisters and brothers. Please add my number in the WhatsApp group.
And the number?
Number I will tell.
Okay, tell.
970
Wait. 97
0
0
40
40
83
83
147.
147. Okay.
Please add.
Even
add
add
the
me
number
also,
in
please.
WhatsApp group immediately, please.
Tell me number.
818
818
4970
4970
616.
616.
Yeah.
Jay, बोलिए.
Is there any special group for BZC?
Add my number. Hello.
Hello.
Tell
Hello
me.
friends, is there any
6.
special group for BZC? 955.
Voice are breaking. Please tell me.
Hello.